Welcome to the deep dive. Our mission, as always, is to take that dense stack of research, the papers, the lab reports, and turn it into something crystal clear, something you can really sink your teeth into. And today, we're tackling something I think we all struggle with. Why is fat so incredibly hard to say no to?
It really is the weak spot in so many diets, isn't it?
Yeah.
Fat isn't just about calories. It has this unique palatability that just seems to override all our good intentions.
Right.
The sources we're looking at today even point out that a preference for fatty foods is, more tightly linked to obesity than a sweet tooth is.
That makes so much sense to me. Sugar is like a quick hit, right? A burst of energy. But fat is. It's the texture, the satisfaction. It's that deep, lingering richness.
Exactly.
Yeah.
The mouthfeel. And that's precisely what makes it so tough to manage.
And this is where our current tools kind of fall short. I mean, you look at the big weight management drugs, the GLP-1 agonists. They work, no doubt about it.
Oh, they're incredibly effective.
But they're a bit of a sledgehammer. They reduce your overall appetite, sure, but they often they flatten the whole experience of eating.
Yeah, you hear that a lot. People just lose the joy of food, which can lead to, psychological issues, or they just stop taking medication.
Yeah.
We need something more precise.
We need a scalpel, not a sledgehammer. A way to dial down the craving for fat without turning off the joy of everything else.
And for the longest time, we just didn't know how the brain did that selectively. We knew the gut could tell fat and sugar apart, but the central command center, how it separates a craving for potato chips from a craving for, say, gummy bears. That was a mystery.
We had found a circuit for sugar, I remember that, an opioid one, but the fat circuit was totally elusive.
Until now.
Yeah.
So today, our deep dive is into a fascinating study by Zheng and colleagues that claims to have finally found it. We're going to unpack this histamine H3 receptor circuit that could, just maybe, be the key to precise weight management.
Okay, so let's start with the big question. If the brain has separate dials for fat and sugar, where are they? How do you even begin to find the exact neurons that only care about fat?
Well, they use this really clever technique. It's called activity-dependent neuronal labeling, using something called FOST-TRAP mice.
FOST-TRAP, okay.
Think of it as a kind of neuronal detective kit. It lets you tag neurons that become active and make that tag permanent.
So you can see which cells responded to what, even long after the fact.
Precisely. So here's what they did. They fed the mice a high-fat diet. Any neuron that lit up in response got permanently tagged with a red marker, like a little I respond to fat flag.
Okay, so the fat responders are now glowing red forever.
Right. Then, after a little while, they gave the mice either more of the high-fat diet or they switched to a high-sugar diet. The neurons active during this meal then lit up with a temporary green marker.
Ah, so you can compare. If a neuron is yellow, it responded to both. If it's just red, only fat, and if it's green, only sugar.
You got it. And what they found was what the paper calls a striking separation.
And where was this separation?
In a brain region called the paraventricular thalamus, or the PVT. That was the hub. They saw these distinct clusters of red neurons and green neurons right next to each other but separate.
That's the key, right, the separation. Because I saw they looked at other big-name hunger areas, the lateral hypothalamus, the VTA.
And in those places, it was all mixed. The fat and sugar neurons were largely overlapping yellow. It was really the PVT that was acting like the primary sorting station.
Okay, so they found the spot, but just because a neuron lights up doesn't mean it's actually in control. It could just be watching.
Exactly. So the next step was to prove they were actually pulling the levers. They needed to turn these neurons on and off themselves.
And that's where chemogenetics comes in. The whole D-DRED-Ds thing, this sounds super technical. Break it down for us.
It's basically a remote control for neurons. DRED-D stands for designer receptors exclusively activated by designer drugs.
A mouth.
It is. But think of it like this. They genetically installed a special kind of molecular switch only into those red fat-sensing neurons. One switch, HM3D, was an on button. The other, HM4D, was an off button.
And a special drug is the only thing that can press those buttons.
The only thing, so they could selectively activate or silence just the fat-sensing neurons and see what happened.
And what did happen?
The results were crystal clear. When they pushed the on button activated the PVT fat neurons, the mice specifically ate less of the high-fat diet, just stopped eating it.
And their sugar intake?
Totally unchanged. Didn't touch it.
Wow! Okay. And when they hit the off button?
The opposite. Silencing those neurons made the mice eat more of the high-fat diet. They overate the fat, but again, left the sugar chow alone.
So it's not a general stop eating signal. It is a dedicated fat-specific break.
An independent functional switch. That's the discovery.
Okay, let's just quickly recap this first part for everyone. So first, we've learned the paraventricular thalamus, the PVT, has these physically separate groups of neurons for fat and sugar. Second, using that dread remote control, they proved these fat-sensing neurons act as a dedicated brake on fat consumption.
A very specific brake.
And finally, hitting that brake only affects fat intake, leaving sugar completely alone. So that raises the next question. If these neurons are the gatekeepers, what's their molecular ID? What makes them different on a genetic level?
Right. What's the special access card they're carrying?
Well,
to figure that out, they had to dive into the cell's genetic blueprint using a method called tree path sequencing.
okay.
It's a fancy way of saying they could grab all the active genetic messages, the messenger RNA, but only from the fat-sensing neurons, and then compare them to the sugar-sensing ones.
Looking for the one gene that stands out.
And one just shot at the top of the list, the histamine H3 receptor, H3R. It was way, way more abundant in the fat neurons than the sugar ones.
Wait, histamine, as in antihistamines for allergies and sleep. That histamine...
The very same.
How does a receptor involved in keeping us awake and dealing with allergies become the master switch for fat intake? That seems really random.
It does at first. But if you think about histamine's role in the brain, it's about arousal, vigilance, and immediate pay attention signal. The histamine itself comes from a little nucleus called the TMN, and this whole TMN to PVT circuit seems wired for a rapid response.
An instant check on what you're eating. Which leads to the next question. What is it checking for? Is it waiting for the calories to hit the bloodstream? Or is it reacting the second the food hits your tongue?
They check that too with fiber photometry and let you watch neurons fire in real time. And they found these PVT fat neurons lit up at the very onset of the mice eating the high-fat food.
So it has to be orosensory, taste, smell, texture.
Texture seems to be the big one. And to prove it, they designed this beautiful texture experiment.
Right, the xanthan gum.
Yes, xanthan gum. It's a food additive that just adds viscosity, that creamy, fatty mouthfeel, but with basically no calories. They mixed it into standard mouse chow of different concentrations.
And what they found is just amazing.
It's stunning. As they increased the xanthan gum, making the food feel richer and fattier, the activity of those PVT neurons went up in a graded proportional line.
The brain wasn't counting calories, it was measuring viscosity.
It's all about the sensation. And the final proof was, when they inhibited those neurons, the mice overate the xanthan gum chow. But when they offered sweet chow instead, the inhibition did nothing. The brake is truly texture-specific.
And the wiring diagram of the brain supports this. They traced the circuit back and found the histamine neurons in the TMN get input from a place called the MA5 nucleus.
Which is the brain's processing center for mechanosensation from the mouth, the feeling of chewing, the pressure on your jaw.
It's a direct sensory highway. Chew something that feels fatty, and the brake immediately gets activated via this histamine signal.
Exactly.
Okay, a quick pause to summarize this part. We found the molecular marker for these fat-regulating neurons is the histamine H3 receptor, or H3R.
Right.
This H3R circuit is triggered by orosensory cues, specifically the texture or viscosity of the food.
The xanthan gum experiment proved that.
And the whole thing is wired for immediate feedback from the act of chewing itself. So this H3R is a brake. But this is where it gets tricky. H3 receptors are usually inhibitory. They calm neurons down. How does activating a calming receptor make a neuron more active and suppress eating?
This is the core of it. This is where this study gets really elegant. It's a phenomenon called biased signaling.
Biased signaling.
Right. So first they confirm the basics. Get rid of H3R and the mice eat more fat, add more H3R, and they eat less. The receptor is definitely the brake pedal. The question is, how does pressing it make the neuron fire?
Because normally it would do the opposite. GPCRs, which is what H3R is, usually signal through a pathway called GI, which is inhibitory.
Exactly. But in these specific PVT fat neurons, activating H3R did the opposite. It enhanced the neuron's excitability, it pushed it closer to firing, and they discovered it's because it's not using the GI pathway. It's using a different, less common one, the G12-13 pathway.
So why does that matter? G12-13 GI, it sounds like technical jargon, but you're saying it's everything.
It is everything. Think of the H3R receptor as a light switch in a smart home. In most rooms of the house, most neurons, flipping that switch is wired to the GI pathway, which dims the lights, inhibition.
Okay.
But in this one specific room, the PVT fat neurons, that same switch is wired to a different system, the G12-13 pathway. And in this context, that system turns up the power to the fat brake. It's the specific wiring that gives it this unique opposite effect.
And they could prove it was only that pathway.
They could. They
Oh.
used a drug called rosin, which
Uh-huh.
specifically blocks the G12-13 pathway. When they added it, the fat-suppressing effect completely vanished. The brake just stopped working.
But the real kicker is the controls, right? They tried blocking other common pathways.
They used blockers for the GQ pathway and the beta-arrestin pathway, and nothing. No effect on the neuron, no effect on eating behavior. It is exclusively the H3R G12-13 axis.
Which brings us all the way back to the beginning, the problem of the sledgehammer.
And this gives us the scalpel. This is why older H3R drugs failed in trials. They were hitting every H3R switch in the whole house, causing all sorts of side effects.
But now we have a precise map. Target H3R, but only in the PVT and only through the G12-13 pathway.
Exactly. You can now design what are called biased ligands drugs that are engineered to flip that H3R switch, but only if it's wired to G12-13. It gives you this incredible precision to just turn down the dial on the sensation of fat without touching anything else.
So to bring it all home, we've established that your preference for fatty foods is controlled by a dedicated brake in your brain's PVT. That brake is marked by the histamine H3 receptor, which responds to the texture of the food and works through this very specific biased G12-13 pathway.
And the evidence that it's all about the sensory experience, the texture, the chewing, is just overwhelming. It seems our brains are incredibly tuned to the feeling of eating fat, maybe even more than the calories it provides.
But that leaves one final fascinating question on the table, one the researchers themselves brought up.
Yeah, this is the next frontier. If this circuit is all about texture and mouthfeel, you have to find a way to completely separate that from the nutritional value.
And the way to do that is something called intragastric infusion.
Right, you deliver the fat directly into the stomach, completely bypassing the mouth. No taste, no texture, no chewing.
And here's the thought to leave you with. If they do that, and this PVT H3R circuit still activates and regulates how much fat the animal wants, what does that tell us?
It would mean that the post-ingestive signals, the gut telling the brain, hey, fat has arrived, are way more powerful and integrated into this texture circuit than we thought. But if the circuit stays silent?
Then it tells us that our deep fundamental drive for fat might be almost entirely about the learned pleasure of that sensation. How much is about energy and how much is about the simple, profound joy of that mouthfeel. That's what's still left to discover.